Let's go on to the next case. That is Prashant Ratnaparki. And Sanjay Godbole, he is your co-author. Okay.
A very good morning to all of you. My name is Prashant Ratnaparki and I will be presenting my topic today, which is association of the risk of cardiovascular complications with a C242T polymorphism in P22 FOX gene in diabetic population. This is a cross-sectional study which is done in Mumbai, and I am not a medical doctor. I am a PhD student. These are the author affiliations, and the authors declare no conflict of interest. The cardiovascular complications can be attributed to three major factors: genetic factors, metabolic factors, and the environmental factors. In the current presentation, I will be basically describing a point mutation which is called as single nucleotide polymorphism. The single nucleotide polymorphisms are basic polymorphisms which occur in a population, and an example is given there, where one particular nucleotide is found in place of the other. Now these are found within structural regions of the gene or can be found in between genes, and in the today's scenario they are all used as biomarkers for health and disease. The gene P22 FOX which we are studying basically produces a protein called as CYBA or cytochrome B alpha chain, which is a component of an NADPH oxidase. Now this NADPH oxidase produces superoxides, and these superoxides are needed for phagocytosis, generally found in phagocytes or endothelial cells of blood vessels. That's how the name comes as PH. The C242T polymorphism is a unique missense mutation. That means instead of histidine, a tyrosine is introduced into the protein at position number 72. Now this is reported to alter heme binding sites of this particular subunit, and because of this affects the rate at which superoxides are produced. Now these are the two references for that. So how do I want to summarize this concept is the P22 FOX, which is a subunit of NADPH oxidase. The NADPH oxidases produce superoxides which oxidize LDL. These oxidized LDL molecules are taken up by monocytes, eventually forming foam cells. Now these foam cells get deposited and adhere themselves onto the endothelial layers of blood vessels, and increasing ROS generates more stress and causes migration of vascular smooth muscle cells, starting atherosclerotic plaques and eventually increasing the risk of cardiovascular complication. So our study basically tries to find out the association between the risk of between polymorphisms in C242T and the use of Framingham risk score in type 2 diabetic patients and the non-diabetic patients. So we had 144 subjects, of which 83 were diabetic and 61 were non-diabetic. The SNP was analyzed using a preformed Tachman assay. Anthropometric measurements were taken using normal methods, and blood biochemistry were determined using automated methods. Now the observations that we got were grouped into two groups. One is called as presence of the C allele and the other is absence of the C allele. That's why we have two cohorts, CC plus CT and versus TT. These are the two cohorts which we are looking at. We also performed t-test, chi-square test. Odds ratios were calculated and the CIs were used after correcting with bonferroni. What we did get is homozygous CC population was 26%, homozygous TT population was 22%, whereas the heterozygous population was as much as 51%. what we did get, which will be seen in the next slide, is the postprandial blood sugar was high in the CC plus CT cohort as compared to TT. So this talks about increasing load of glucose and hence a possible ROS mechanism. Now, most of these parameters which were identified were non-significant when you compare between CC and CT, except for postprandial blood sugar, which is shown here. Sorry. Then we performed a cross-tab analysis using SPSS 21, using Framingham risk score, the state of diabetes. That means code 1 was given whether the patient was diabetic, and code 0 was given if there was no diabetes. And this was identified, and we got a very strong risk ratio of 5.22, which was significant at less than 0.05 value of significance. The CC plus CT cohort had an odds ratio 1.55 as against the TT cohort, whereas for FRS less than 47, now the 47 was taken as mean of healthy subjects plus or minus 2 SD. That's how the value of 47. The values for patients with FRS less than 47 were non-significant, and hence you can see this. So we had 65% of the diabetic population which had FRS more than 47 belonging to CC plus CT cohort as compared to TT cohort. And those who had less than FRS less than 47 were only 18%, although that was statistically not significant. We used a similar cross-tab using FRS, using diabetic status with BMI. We've used BMI as value 25. With this, we found Pearson's value of 4.34, which was significant at 5%. And the risk, odds ratio risk was 1.27 for CC plus CT cohort as compared to the TT cohort. The next, what we did was we used the odds ratio of diabetic status, the genotype, as well as FRS and BMI as 2 layers. So the result obtained was whether you had a diabetic status or no diabetic status, whether you had a C allele or no C allele, plus differentiation between FRS greater than 47, less than 47, and categorization of BMI. In this entire analysis, we found that diabetic population whose FRS was more than 47 had a BMI of greater than 25. These constituted 45% of our population with an odds ratio of 5.6, which was very significant. We also did find that if the BMI was less than 25, the odds ratio was 2.5, and that was found in 30% of the population. The FRS less than 47, the population which had BMI greater than 25 constituted 35% of this population with an odds ratio of 1.24, whereas this particular parameter was non-significant. So what we conclude out of our studies basically is the subjects which we found in our study, those who have a C allele, they have a higher value of postprandial blood sugar. We had 65% of the diabetic population which have a FRS score more than 47 and are at a slightly higher risk of cardiovascular complications. The odds ratio was 1.55. 60% of diabetic subjects with C allele and BMI greater than 25 are also at 3 times the risk for cardiovascular complications. Odds ratio is 1.29. So if we have subjects which have the presence of C allele, that is CC plus CT, and if the BMI values are maintained less than 25, the risk of cardiovascular complications can be reduced. In short, the other way of looking at this particular result is those individuals who are diabetic and also have a TT combination of their genes, they are slightly protected from these damages. We don't claim that that's something which can be depended upon, but that's what our study shows. So if we review our study, the strengths of our studies, this is one of the few studies which actually tries to link C242T polymorphism with cardiovascular complications in Indian diabetic population. And our results are similar to one published in thrombosis research. The limitations is one is a cross-sectional study. Second, we have low participants. So if this study was conducted in large number of patients, then probably this study would be a little more better. I acknowledge these people, and I also acknowledge IDEC 2018. Any
A
questions?
good case, a good study, but the only thing is the conclusion is that you only can identify the risk factor for those who develop diabetes irrespective of the BMI. You found that the BMI on the higher side, more than 25 and less than 25, with the C allele, the risk was higher,
Yeah.
right?
Yeah.
Irrespective of BMI.
Yes.
So the BMI cutoff doesn't give benefit.
No, actually the BMI cutoff, if you have BMI less than 25, the risk ratio comes down. Can I? So if you have the BMI values of less than 25, the risk ratio is 1.05, which is as good as equal. And so the whole point is if BMI is maintained, the genetics of an individual may not be controlled. That's what you inherit. But BMI is one environmental factor which you can control and cut down the risk of cardiovascular
Which
complications.
chromosome was this?
So this was chromosome number 6, and it has 6 exons, this particular gene.
Short, long arm,
So the long arm.
long arm, 6 chromosome.
Yeah.
Anybody has any questions? Yeah.
I would like to know why you have combined these two cohorts, heterogeneous as well as homozygous. Why not separate?
So there are 2 reasons. One is the numbers are extremely small for a CC and a TT comparison. Second is we are trying to look whether the presence of a particular allele either in homozygous or a heterozygous combination actually contributes to the risk. So that can be done by, and most of the papers have actually done this for the same reason.
Because separating them, I think, will be a better idea.
Yes, I agree to that, but
But
then
he
we have,
explained the number of patients is small.
we really need very large number of patients to make some statistical study for that.
I think this is just
The thing is, the word diabetic, if you are going to publish this, I mean, since you are a non-diabetic, non-diabetic,
non-doctor.
person, I think that word diabetic should be taken out.
Right, sir. I'll take it.
I think as a research is a good one. I mean, you know what is the genetic cause for the people who develop cardiovascular disease. And each kit he told me cost 65,000 rupees. And that has been repeated twice or thrice. And it's a large study. I think you go ahead and do this study in a larger scale and maybe next year present further data on this.
Right, sir.
Okay, it's a good work and
Thank you,
continue doing the good work.
Thank you,
Yes.
sir.